A client came to us mid-campaign, GAL-NAC conjugate, liver target, solid data, clean IND package, then asked one question, what do we do for the CNS indication? That's the moment most programs stall. The liver is solvable. Everything beyond it is where precision therapeutics get genuinely hard. And that difference between a conjugate that works and one that doesn't come down to decisions made very early in development. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, where we help biotech and pharma teams build, characterize, and advance their peptide oligonucleotide conjugates from concept through regulatory submission. What are peptide oligonucleotide conjugates? Peptide oligonucleotide conjugates are hybrid molecules, two distinct chemical languages joined deliberately. The oligonucleotide component, your siRNA, ASO or antisense sequence, does the molecular work, silencing a target gene, blocking a splice site, editing expression at the source. The peptide is the navigator. It carries the cargo to the specific cell type, crosses the membranes that would otherwise reject a marked nucleic acid, and determines whether your molecule reaches its site of action or not. Without the peptide, you have an oligonucleotide. With the right one, you have the targeted medicine. Why these conjugates matter? Here's what's driving serious interest in this modality. Most approved oligonucleotide drugs work in the liver because that's where passive delivery concentrates them. GalNAc conjugation solved the hepatic targeting problem elegantly, but your patient population may not live in the liver. Muscle disease, CNS disorder, oncology, these require delivery beyond where passive accumulation can take you. Peptide conjugation breaks that boundary. It increases intracellular delivery, reduces the dose needed to achieve silencing, and cuts the off-target exposure that raises toxicity questions at the FDA. For any program trying to expand beyond hepatic indications, this architecture isn't the optional, it's required. Two main synthesis strategies. The manufacturing question arrives early and shapes everything downstream. There are two primary synthesis strategies. In stepwise solid phase synthesis, the peptide oligonucleotide sequences are assembled on the same resin, a single continuous build with tight structural control. The demand is that protecting group strategies for both chemistries must coexist without conflict, which is harder than it sounds at scale. The alternative is parallel synthesis. Purify each fragment independently, then conjugate them in solution. This lets you optimize each component separately and pivot on one without rebuilding the other. The trade-off is a conjugation step that introduces its own yield and purity variables. What a less experienced team misses is that this isn't primarily a chemistry decision. It's a program decision. Your timeline, scale targets, and regulatory strategy all inform which path is defensible before an agency. Midpoint real world scenario. We worked with a team developing a splice switching ASO conjugated to a cell penetrating peptide for a muscle indication. They come in with a stepwise protocol that had performed cleanly at cleanly at microgram scale in a research setting. When they transferred to GMP compatible conditions at 100 milligram, protecting group orthogonally broke down at the coupling step between the lysine residue and the phosphate backbone. 3 months and 2 vendors later, they moved to a parallel synthesis with a redesigned linker architecture, and that version entered IND enabling studies. The chemistry wasn't wrong here, the strategy had never been stress tested at the scale. The power of smart linkers, which brings us to the linkers. Linker chemistry is where conjugate programs succeed or fail quietly. The linker determines whether the conjugate stays intact in system circulation, releases the oligonucleotide payload at the right intracellular department, and survives the analytical conditions used to characterize it. Cleavable linkers, disulfide bonds, pH-sensitive chemistries, enzyme-labeled sequences are designed to release the payload after cellular uptake. Stable linkers maintain conjugate integrity through delivery. The choice depends on your mechanism of action, target compartment, and how your molecule behaves in endosomal conditions. This is where the gap between published literature and manufacturing reality is most pronounced. Peptides improve cellular delivery. The peptide component carries its own set of considerations. Cell-penetrating peptides, receptor-targeting sequences, fusogenic peptides. Each class interacts differently with the target cell membrane, the endosome, and ultimately the site where the oligonucleotide needs to act. Peptide selection isn't just about binding affinity, it's about endosomal escape efficiency, serum stability, immunogenicity risk, and where the peptide introduces a novel characterization button into your CMC package. Analytical testing and quality control. You cannot file without the data you can defend. Mass spectrometry confirms the molecular identity, conjugation site, and absence of unconjugated fragments. HPLC delivers the purity profile and flags sequence variants that could surface as impurity questions during review. Impurity profiling for oligonucleotide conjugates is structurally more complex than for small molecules. You are characterizing a hybrid entity with multiple distinct degradation pathways. Strong analytics don't just support the science, they protect the submission timeline as well. Future trends and clinical potential. The clinical pipeline for this modality is accelerating. CNS programs, rare muscle diseases, and oncology indications are all in active development. Regulatory precedent for peptide oligonucleotide conjugates is still being established, which means the teams building rigorous analytical and manufacturing packages today are the ones that will move through review without the extended back and forth that cost programs entire quarters. Peptide oligonucleotide conjugates represent a genuine expansion in what nucleic acid medicine can reach and how precisely they can act. The science is mature enough to move into the clinic. What separates programs that get there from the one that stall is depth of chemistry, analytical, and regulatory strategy behind them. If your team is evaluating this modality at any stage, we are glad to have that conversation. Not a detail, but we just want to understand what problems, challenges in this modality you are facing. Thank you so much for watching this session, and see you in the next one. Until then, goodbye for now.